today's lecture, we'll discuss how plasmids make, what is meant by plasmid, what are different characteristic features of plasmids, how these plasmids transfer from one bacterium to another, what is the role of plasmid in transformation. This we'll see in detail. We know well plasmids are a small circular DNA just like a bacterial DNA. So, this differ from bacterial DNA. They are small, autonomous, self-replicating bodies which are present in the bacterium. As far as structure of this plasmid is concerned, so plasmids, they are circular, self-replicating. As far as characteristic features of these plasmids are concerned or functions of these plasmids are concerned, how restriction enzymes work if we want to cut this DNA, how restriction enzymes work with this DNA, how the role of ligase, all the answers we'll see in today's lecture. So, with the help of certain animations, we'll see in detail the structure, characteristic features and role of plasmids as far as genetic engineering is concerned. Nowadays, these plasmids are very important. It plays a very important role in genetic engineering, formation of rDNA technology or recombinant DNA. So, we'll see in detail now plasmid. So, a plasmid is a small self-replicating circular DNA molecule found in bacterial cells and some other organisms. Plasmids are separate from the bacterial chromosome and can replicate immediately. So, you just see this is the structure of bacterial DNA. So, here this brown color, that is bacterial DNA, wherein this blue color highlighted structure is nothing but the plasmids. So, this plasmid is a circular, self-replicating, small molecule found in bacterial cells and some other organisms. As far as characteristics of plasmids are concerned, so plasmids are autonomous replication. So, it is having characteristic features of autonomous replication. So, plasmids can replicate independently. So, plasmids are having capacity to replicate independently of the bacterial chromosome. So, it is having another characteristic feature called self-contained. So, plasmids carry their own origin of replication. Plasmids, they are having their own replication origin which allows them to replicate. In addition to that, we know well, this is a very popular statement that plasmids are circular DNA. So, plasmids are typically circular, double-stranded DNA molecule. In addition to that, plasmids are smaller in size. So, they are having small size structures. So, plasmids are usually smaller than the bacterial chromosome. In addition to that, one more very special characteristic feature of plasmid, that is they are accessory genes. So, plasmids often carry genes that provide additional functions such as antibiotic resistance. So, plasmids, they carry genes that provide additional functions such as antibiotic resistance or virulence factors. In addition to that, there are certain functions of plasmids we will see in detail. So, plasmids, they are having antibiotic resistance. It means that plasmids can carry genes that confer resistance to antibiotics. In addition to that, one very important function related with the plasmids, that is virulence factors. So, plasmids can carry genes that contribute to bacterium's ability to cause disease. So, it is nothing but the virulence factors. Metabolic functions are also observed in plasmids. So plasmids can carry genes which are involved in metabolic pathways such as nitrogen fixation and biodegradation. So this nitrogen fixation is very important as far as functions of plasmids are concerned, and in addition to that, biodegradation also perform, or it is very useful, or plasmids are very useful in the process of biodegradation. In addition to that, one more function, that is gene transfer. So plasmids can facilitate the transfer of genes between the bacteria. So from one bacteria, it is able to transfer to the another bacteria. So this is one more very important functions of plasmid. In addition to that, as far as types of plasmids are concerned, so the first one, that is nothing but the conjugative plasmids. So see here, these conjugative plasmids can transfer themselves from one bacteria to another bacterium through a process called conjugation. So here in this figure, you can see here, this is the mobile plasmid. So this cell, this is the bacterial cell. From here, the transfer of one gene, and it is going to transfer to the another bacterium, which is called as recipient bacterium. So here this is the donor bacterium, and here this is the recipient bacterium. So it means that, see here, so this plasmid is going to transfer from one bacteria to another, means the bacterial bacterium can transfer, or the plasmid can transfer from donor to the recipient. So resident, that is antibiotic, that is in red color, and susceptible antibiotic, that is in green color. So you can see here resistant antibiotic B and susceptible antibiotic A. So this process is performed with the help of conjugation process. So this conjugation process, here formation of conjugation tube within the bacterium, and it is going to easily transfer from one cell to the another cell. So this bacterium which is having plasmid, that plasmid can transfer from one cell to the another cell. So transconjugants, so here resistant antibiotic A and resistant antibiotic B. So experimental characterizations are concerned. So plasmids, as far as size, here sequencing and replicons, PCR, we are taking help of PCR, and they are resistant genes which can be performed subsequently with the help of polymerase chain reaction, that is PCR. Second type, that is non-conjugative plasmids. So this non-conjugative plasmids cannot transfer themselves, and truly an other mechanism such as transformation or transduction. So here the second type, non-conjugative plasmids, here we cannot observe conjugation tube, but here natural transformation takes place. So here bacteriophages, this is the free DNA, and the process called as transduction. So with the help of transduction, the movement of plasmids from one cell to another cell. Third type, that is episomes. So plasmids that can integrate into the bacterial chromosome. So here see, this is the cell. Here we can see clearly cytoplasm. This is lana, and this is nucleus. This is the chromosome, and here this is the episome. So this episome is attached with this chromosome. So this orange color structure, that is the episome, which is attached to the plasmid. Okay, so this is plasmid. Second, self replication. So here one cell of this particular episome, and here you can see it becomes two. So self replication takes place here. So replicated chromosome. So likewise you can see here this chromosome contains episome, and this chromosome also contains episome. So from one bacterium, from one cell, we are getting two cells, separate cells along with the plasmids. So this is segregation. So from one particular cell, so this episome replicates, self-replicate, it forms the replicated chromosome. So from one cell we are getting two cells here. We are getting two cells. In the screen it is very clearly mentioned about replication and segregation. We are getting two different cells. In addition to that, viral plasmids are also observed. So these plasmids that are derived from viruses and can replicate independently. So see here, this is the A diagram and this is the B diagram. So here in A, this is green color, that is the virus or viral plasmids. So these viral plasmids deliver to host or viral packing cells, and we are getting more viral cells. So more viruses here we are observing. So second thing, modified viral plasmids. So here all these are small modified viral plasmids, and we are getting viruses. Same thing we'll discuss with the help of this animation. So a plasmid is a small circular piece of DNA found in bacteria. So this is a bacterial plasmid, and we'll see what to do with this plasmid, how restriction enzymes work, how restriction enzymes are helpful to cut the DNA, how we can incorporate this cut DNA, which is also called as a recombinant DNA, into the linear DNA. So see here, the plasmid encodes genes such as resistant to the antibiotic called ampicillin. So here we are having three different regions, out of which the plasmid has also a region called origin of replication, where protein synthesis is initiated. So this is the origin of replication. This much region is called as origin of replication. In addition to that, a third region is called lag Z region. So here this is the lag Z region. This also having certain regions. So within the lag Z region there is a high concentration of restriction sites. So each of these is a unique location where the plasmid can be cut by a restriction enzyme. So here role of restriction enzyme is very important. Now see here, a restriction site contains a sequence of nucleotides that is recognized by a restriction enzyme. So this yellow colored structure highlighted, and these are the nitrogen bases, and these are the nitrogen base pairs. C joins with G with the help of triple bonds, wherein this T joins with A with the help of double bonds. So here C joins with G, G joins with T, A with T, and G with C. So this much region is nothing but the restriction site. So on this restriction site, you can observe here, each restriction site has a unique sequence of nucleotides. We know well, in any nucleotide which contain nitrogen base, pentose sugar, and phosphate group. So here it represents a nucleotide. So each restriction site has a unique sequence of nucleotides that is recognized and cut by a specific restriction enzyme. So here you can clearly see this is the restriction site where actual the DNA is going to be cut. So the cut plasmid has sticky end. After the completion of activity of restriction enzyme, so DNA is cut or this plasmid is cut and it forms the sticky end. So here this region is called as sticky ends. This region is called as sticky end, where the broken nucleotide pairs have a slight charge. This makes them attracted to binding with a complementary sequence and reforming hydrogen bonds. So development of hydrogen bonds or formation of hydrogen bonds. In many molecules of a single restriction enzyme are added to a test tube of plasmids. So the product is thousands of copies of a plasmid cut in the same location. So we are getting similar size plasmid, thousands of copies of plasmids with similar size, or restriction enzymes are able to cut on the same location, and we are getting thousands of copies. The next step of gene cloning is to prepare the DNA extracted from the organism that contains the gene of interest which we want to locate. This step is very important. Just like the plasmid DNA, this DNA also has restriction sites. So, where exactly we want the cut, likewise like the DNA, in plasmid also we are getting the cut. The goal is to cut the DNA into gene size pieces, means smaller size pieces or gene size pieces we are going to get with this. The cut plasmids are then mixed with the gene size pieces to linear DNA in hope that they will bind with one another. Since both the plasmids and linear DNA were cut with the same restriction enzyme, the nucleotide sequences of their sticky ends are complementary. The slight charge of the sticky ends, which I have already mentioned. So, I repeat, the slight charge of the sticky ends of the plasmid and the linear DNA attract the pieces to one another and it is going to join. So, as a result of which hydrogen bonds form between the nucleotide pairs. The weak hydrogen bonds holds the DNA pieces together long enough for ligase to bound DNA phosphate backbone together. So, here the newly added enzyme, that is ligase, which bound the DNA's phosphate backbone together. Hopefully, a plasmid will bound with one of the gene size pieces of linear DNA. This is now considered recombinant DNA because it is a DNA in new combination. So, earlier we have taken the DNA and here we have added piece of DNA, newly added DNA, which is also called as a recombinant DNA. As this DNA multiply, the same time the added DNA or the recombinant DNA also get multiplied, and this is the benefit of plasmid DNA.